Hello and welcome to Biology 6. Okay, so you can see I'm trying to sort of change the formula around a bit. Um, I'm not really, I don't really want to draw the pictures anymore. I don't think that's, yeah, it's not what I want to do, so I'm going to go with this kind of simple vector approach for now. Okay, so we've got the vesicle here, as you can see, it's denoted by this ball here. This is the plasma membrane. Um, So I'll just put a description similarly to what I did in the last episode. So the description will be um, a small fluid filled sac in the cytoplasm. Um, Surrounded by a membrane. So yeah, so anywhere I do a line and not just the fill, then there's going to be a membrane. And a lot of stuff does have membranes. Um, okay, so this is kind of transportation organelle, I guess you could say. Um, um, so it transports substances in and out of the cell. Yeah, my handwriting's not too great today. Um, um, so some are formed by the Golgi apparatus or the endoplasmic reticulum. We're going to be moving on to the Golgi apparatus in a minute. Um, so some of them, so some formed um, by one it's not quite getting the pressure right. I may just put more pressure. Well, one will go the apparatus or to the endoplasmic um, reticulum. And others just formed at the surface. All right. Okay, so here we can see the proximity of the vesicles with its object here, which is the Golgi apparatus. Um, and yeah, and vesicles. So just to continue from that, um, yeah, so I put vesicle there. I haven't actually labeled here. Um, so yeah, the vesicles are often in close proximity to the Golgi apparatus. Um, yeah, so description. I wonder what shorthand for description is. I might have to look that up at some point. Is a group of group, so it's a grouping, fluid filled flattened sacs. Like I mentioned, vesicles often so there we go. That's fairly clear. Yeah, I think in times when my handwriting's not so good, I need to focus on clarity. So it would be good way to retain parity. So what's the next thing then? Yep. So it's going to process. Okay. So it does processing and packaging of lipids. Okay. Listen, if I'm trying not to go too low here, got the proteins there. So, yep. And also makes lysosomes. Lysosomes, as you might recall, a round organelle surrounded by membrane with no clear internal structure, and does contain digestive enzymes. So lysosomes are good for breaking down worn out components of the cell. Okay, so this Golgi apparatus is where the lysosomes come from. So it does process and package lipids and proteins, and makes lysosomes for lysosomes, the round organelles, good for digestion. And what else can I talk about there? Not much, not much. And now I thought I'd vary it around. I could have labelled this one as well, but I was wondering if I was just curious whether I'd actually prefer putting the arrows on here or doing it beforehand. So yeah, this is, like I say, this is very much work in progress. I think with biology, I kind of didn't anticipate everything that the really how much it was going to be reliant on pictures, which perhaps I should have done, but I think I'm going to have to do this a lot more because I think a picture that I do beforehand is certainly going to turn out better than something I do freehand. And I think that is kind of the kicker in terms of what I want to do. So yeah, and I think in future I will do all the labeling beforehand as well. I don't know actually. I don't know. Then we have Krista and Matrix inside. So Krista, matrix. So I guess it's the cell matrix, which is the goo, and then the Krista is the dot. I'm not sure though. Now, maybe it's not quite like that. So this is obviously mitochondrion. But yeah, one thing I was thinking of doing for biology 8 is to pre-do all these images beforehand. Because the good thing is once I've got like a mitochondria and a vesicle and all the other organelles, I can perhaps adapt them, do things with them and stuff like that. So that'll be what I'm thinking about. And yeah, so mitochondrion has got certain qualia. So okay. Usually oval shaped. Double membrane, of course. So the inner membrane is actually folded. This is the thing about crystalline matrix. I'm going to need to look this up a bit more. So the inner membrane is folded. Okay. And I think the structure is the crystal. Structures. I think next time make it a bit bigger. I've got a bit of space to the right, so I could have done that. I'm not going to do it now there. So the structure is created. Inside the crystal is the matrix. Yeah. So I think the crystal is the whole structure. And then perhaps those little dots are some kind of enzymes. I'm going to clarify this though. I really want to try and get on top of this stuff. It's a matrix. And the matrix contains enzymes. The matrix contains enzymes. Respiration enzymes. Respiratory enzymes, maybe? Okay, so the key thing about it is where ATP. So that's what's that? Adenosine triphosphate. Yep. I'll just put ATP. I will write it down, but with the amount of space I've got at the moment, it's probably not the best thing. So the mitochondria are the workhorses. So we'll just put found in large numbers. In very active cells. So sort of high energy cells are going to find a lot of mitochondria. And that, I think, is going to be it for the organelles I'm going to do in this video. So yeah, so the plan of action, following along, is I'm going to go through a few more organelles, and then in episode 8, I'm going to try and do a bit more work and try and get some, yeah.